Good morning, everyone. Thanks for attending. It's great to see everybody's faces. It is my great pleasure to introduce our grand round speaker for today, Conor Liston. Conor is professor at Weill Cornell Medical School, and initially started his training at Harvard, and an undergraduate went on to do get his PhD and MD from Rockefeller and Weill Cornell, did a postdoc at Stanford, and then has been at Cornell ever since, doing amazing work. He's been focusing on prefrontal cortical circuitry and its role in psychiatric disorders, OCD, depression, and basic work in that area. And you probably see his articles coming out in Nature and other very high visibility journals very often. He had a very important, impactful paper recently in Nature about circuitry and depression. And I've been along and admire his work. And I recently had the humbling experience where I was talking with Conor about neuroimaging work we were doing about the importance of focusing at the level of the individual and getting lots of measurements in an individual, as opposed to trying to get 1,000 people in a data set and learning about circuitry. And I said, like, excitedly, we're getting 3 scans in everybody now. He said, that's really, really great. We're getting 40. So it was humbling, but it just tells you the quality of the work and the level at which he's operating. And very pleased that we have the opportunity to have him here. I hope many of you will get a chance to meet with him and benefit from his outstanding perspective and really, I think, very important way of pushing forward the envelope and where we've been stuck for many years. Conor, thank you for being here.
All right. Thank you, Andrew, for that very generous introduction, and thanks for inviting me here. I'm really glad to be here. Fun to reconnect with old friends and hopefully meet some new people too. So just by way of disclosures, I'm a scientific advisor to Delix Therapeutics and Brainify, but I won't be discussing any work related to my advisory work with these organizations today. So what I do want to discuss today are a series of efforts underway in my lab that are aimed at understanding the mechanisms that control mood state transitions in depression. Depression, we know, is this fundamentally episodic psychiatric syndrome. It's defined by periods of low mood interposed between periods of wellness. And we've learned a lot from the dominant approach in the field, I would say, which is to study depression in a cross-sectional way, compare a group of depressed people with a group of never depressed healthy controls. But if what you want to do is understand the mechanisms that control these kind of dynamic switches over time, as Andrew alluded to, what we think we need is more longitudinal work, perhaps in smaller groups of individuals at first, that track these switches as they're occurring. And a really beautiful example of this work is shown here. I could find very few examples quite like this. This is from Bob Post and his colleagues over 20 years ago. They just tracked a small number of patients with bipolar depression or unipolar depression and described what they saw. And so what you see here, these are 3 individuals. Below the x-axis is depressed mood. Above the x-axis is hypomania or mania. And 0 is kind of euthymic normal mood. And one thing you can appreciate just by glancing at these 3 individuals is that they look very different. Like this one has kind of characteristic durations to their depressive episodes. This person's episodes are much shorter. They switch back and forth with higher frequency, and they have relatively fewer periods of sustained wellness. And the mechanisms that explain these individual differences and that determine why a person gets depressed, today as opposed to last week or next month, and when they get depressed, what determines how long they stay depressed. And then conversely, when you treat them and they get better, are there mechanisms that are involved in kind of initiating that recovery and perhaps others that are involved in sustaining the remission over time? We really don't know the answers to basic questions like that. And so what we've been trying to do in mice is serial 2-photon imaging, before, during, and after chronic stress and after antidepressant treatments, and in humans, serial fMRI imaging to try to get a handle on what is changing in the brain in association with these switches in mood. And that's what I'm going to tell you about. I have some extra slides built in today. I'll probably skip over some of them, but we can return to them if we have questions about them. Undoubtedly, many mechanisms are involved, but I think converging evidence suggests that one of them might involve the remodeling of these structures called postsynaptic dendritic spines. For people unfamiliar with spines, they're microscopic protrusions on the dendrites of neurons. Most spines contain synapses. I'll be using these terms kind of interchangeably over the next few minutes. Not all spines contain synapses. We can discuss that, but we've known for some time that chronic stress causes a loss of spines and synapses in the prefrontal cortex and in other brain regions, and that antidepressants like ketamine tend to do the opposite, causing synapse growth. And so it's been tempting to speculate that the growth of new synapses after antidepressant treatment might be causally involved in initiating that switch. But of course, it's also possible that they're nearly correlated. And that's a question that has more than just academic implications. If we think that synapse formation is causally involved in driving a switch in mood state, then other interventions aimed at boosting that mechanism might be useful as treatment. So we really wanted to get a handle on that. And that's the first series of studies I'm going to describe. This is led by three really talented lab members: Rachel Moda-Sava, who's now at Harvard; Mitch Murdoch, who's now a postdoc at MIT; and Pooja Parekh, who now has her own lab at the University of Texas. Check out Pooja's work. They set out to ask these three questions. First, does ketamine, this rapidly acting antidepressant, does it selectively rescue synapse loss in the prefrontal cortex? Second, how does the remodeling of synapses influence the function of the circuit? And then third, are they causally related? And I won't bury the lead. I'll tell you that we found that the formation of new synapses was causally important, but not in the way we expected. I'm going to try to unpack that over the next few minutes. So before I show you the data, just to orient you to the method we're using here, we implant a microprism into the brain of the mouse that affords optical access to the medial prefrontal cortex in the living brain. And using the contours of the prism and vascular landmarks, we can return again and again to the exact same position with micrometer precision. You see an example of that here. In these mice that are expressing yellow fluorescent protein on a Phi1 promoter. So they're mostly layer 5 and some layer 2-3 neurons. And by returning again and again to the same areas, we can ask what effect stress is having on the remodeling of spines and how ketamine in turn modulates that. So showing you kind of the first experiment they did, implant a prism, give the mice some time to recover. Then we do 2-photon imaging before and after various chronic stress manipulations. The one I'm going to be showing you here is chronic corticosterone, kind of the mouse version of cortisol in the drinking water. We see this with other stressors as well. And then you just asked what impact stress had, or CORT in this case, on the elimination of spines, which we quantify like you see here, by counting spines that are present in the first image, but not in the second, like these arrows, or spine formation, spines that are present in the second image, but not in the first. And what they found was a big increase in spine elimination. That's perhaps not surprising based on what I just told you we already knew from the fixed tissue studies, but because we're doing this in vivo, we can begin to ask more targeted questions like whether there's a logic to the spines and the synapses that are being lost after chronic stress. And we found that there was, and we did that in various ways. I'll show you one here. We know that one of the functions of dendritic branching is to segregate functionally homogeneous synaptic inputs. So in other words, connections from region A will tend to synapse on branch 1, but not branch 2, whereas connections from region B will tend to synapse on branch 2, but not branch 1. And what we found was that the distribution of lost spines was highly bimodal. You can see that here. About half of branch segments showed spine loss rates comparable to controls, and about half showed markedly elevated spine loss rates, sometimes up to 10-fold the normal number in controls. We also found that the lost spines were spatially clustered. They tend to occur together in groups on branch segments, suggesting that particular projections are being targeted for pruning, perhaps, and we're trying to follow that up now. We then treated the same mice with ketamine or a vehicle control and then imaged them a third time a day later. Anyone who works with ketamine in the room will know that this time point was chosen because many patients with depression experience a pretty strong recovery a day after the treatment. And so we wanted to ask what impact ketamine was having on spine dynamics at that time. And we see basically the opposite of what I just described with chronic stress. a formation of new synapses, and that the distribution of those new synapses and spines is bimodal. It's affecting mainly a particular set of branch segments, and it's also spatially clustered. And so that led us to ask then whether it might be the case that ketamine is acting in a targeted way to restore lost synapses to the same position that they used to exist prior to the chronic stress. And so to test that, we classified each spine that formed after ketamine treatment as a restored spine if, like this blue one, it formed in a position where a spine used to exist and then disappeared, or a de novo formed spine was one like this purple one that formed in a position where we didn't observe a spine in either of the previous two images. And what we saw was a big boost in spine restoration, much more than you'd expect by chance based on the observed rate of spine formation. And that's kind of the glass half full view. The glass half empty view, which I want to just emphasize because I'm going to return to it in a few minutes, is just, you can appreciate it by taking another look at the same data. This is about half of the spines that form after ketamine treatment are restored spines. And that means, that about half are not. And doing some math, we find that although the animal is behaviorally normal, so to speak, it looks like it had never been stressed. I haven't shown you that, but I will. And the number of spines is now back to kind of unstressed control levels. The configuration of spines is only partially restored. And we think that might be relevant for the durability of the effects. And I'm going to return to that in a couple minutes, so hold that in mind. Shifting gears, we next asked about the functional correlates of these changes. So this is the same experiment, imaging before and after chronic stress and again after ketamine treatment. But instead of imaging spine remodeling, we're looking at circuit function using a genetically encoded calcium sensor, GCAMP6, that's expressed on a synapsin promoter, most neurons in this field of view. And one of the predictions you might make if many synapses are being lost is that the kind of structure of correlated activity, functional connectivity between neurons in this field of view might be altered. And that's something we can compute just by looking at correlations in the kind of spontaneous calcium transients we see in these mice. These are, by the way, awake, kind of resting mice, so to speak. They're not engaged in a task. We do have them in other experiments engaged in tasks. I'll show you a couple of examples of that in a few minutes. And so what we found consistent with that prediction was a loss of correlated activity. This is depicted in two different ways here. These correlation matrices, warm colors depict pairs of cells that are strongly functionally coupled. And you can see no fancy stats needed. Just by glancing at this, there's a loss of warm colors after chronic cord administration in this representative mouse. Another thing that we observed, which we didn't necessarily anticipate, but kind of stood out to us, you can see in these raster plots. Left to right is time. This is about 6 minutes of imaging. Each row is a cell, and the elements are colored based on how active the cell is at a given time point. And what you can see in the baseline condition is that most of the activity that occurs in this field of view occurs in the context of these multi-cell ensemble events where many cells, or even most cells in the field of view, become active at about the same time. And after chronic court treatment, the occurrence of these ensemble events is reduced in frequency. And when they occur, they tend to involve fewer cells. And we find that ketamine rescues both of those effects. So we've seen these effects of ketamine and stress on spines and on circuit function. What about behavior? Are they behaviorally relevant? So we think that they are in a number of ways. I'm not going to walk you through most of these, but check out some of this work if you're interested in understanding this further. I will show you two examples. One involves motivated escape behavior in response to threats, and the other is like effortful reward-seeking behavior and effort-based decision-making. And I'll show you that one first. So this is really nice work from a former MD PhD student in the lab, Rob Fecho. Rob adapted a task that had been extensively used in rats to study effortful reward seeking in this task, which he adapted for use in mice, automated it, made it kind of compatible with physiology. The mice learned to navigate left or right in a T-maze to obtain a large reward or a small reward. And to get the large reward, they need to climb over a barrier. And we can couple the large reward with barriers of various sizes, and we can adjust the ratio of the large reward to the small reward to quantify like how much effort a given mouse is willing to expend to get a larger amount of reward. And in this task, Rob was measuring through an optical fiber ensemble events in anterior cingulate medial prefrontal neurons that project to the nucleus accumbens. And summarizing a lot of work, what Rob found was that these accumbens projecting cingulate cells encode a reward predictive signal that scales with effort. So it's sort of integrating information about efforts and rewards. And when you optogenetically silence these cells, and again, this was not exactly what we expected, but when you optogenetically silence these cells after the mouse has already made its decision and expended effort, but before it gets the reward, you will reduce the likelihood that the mouse will make a high effort, high reward choice on future trials. In contrast, if you optogenetically silence in the kind of run up to the decision point and during the decision, that had actually no effect, which was not what we expected, but the data were very clear. It seems that this signal is required, for integrating information about effort and reward and reinforcing future decisions to expend effort to get these rewards. And in chronic court-exposed mice, their high effort, high reward choosing dramatically falls. They go for the low effort, low reward choice almost every time. And this signal is disrupted. And in the animals that show the biggest disruption in behavior, we see an almost complete silencing of this signal. So we're currently looking at how ketamine influences the encoding of effort and rewards in this circuit, but we think this is one way that these ensembles might be important. Okay. Shifting gears to the question I posed at the beginning, the third one about how these things are related in terms of cause and effect, I'll show you two experiments that we did to kind of unpack that. One is, if you think A causes B, A should come before B, and if instead A comes after B, tells you something about cause and effect. So we just wanted to ask what's kind of order of events here in terms of spine formation, changes in behavior, and changes in circuit function. The behavior that we're looking at here is like active avoidance behavior in the tail suspension test, kind of a measure of how a mouse responds to threats. And what we see is that the effect on immobility in the tail suspension test is very rapid. It's detectable in 3 hours. It's actually, in later work, we found detectable much more quickly. And if you look at the spine formation effects, the answer was very clear, not what we predicted, but the formation of new spines is slower. It's not detectable until 12 hours. It peaks after 24 hours. And so that told us right there that the formation of new spines in these, at least in these Thi1 expressing neurons in the prefrontal cortex, could not be required for initiating ketamine's effects on this behavior. We've seen this in other behaviors too. But one hint for an alternative role for these new spines and new synapses came from this scatter plot where we saw that the restoration of lost spines, and you'll recall restored spines were operationalizing as those spines that popped up in a position where we saw a spine before stress and then that spine disappeared. We found that the restoration of lost spines measured at 2 days or 1 week was highly correlated with the maintenance of ketamine's antidepressant effects on behavior in this assay. And so we wanted to ask whether these new spines might be required for maintaining ketamine's antidepressant effects. And to do that, we teamed up with a group at the University of Tokyo led by Haruo Kasai and Haruhiko Bito. They developed a synaptic optogenetic tool, which they call activated synapse photoactivatable RAC1. The name is a bit of a mouthful, but the tool is very simple and beautiful and quite elegant. It's a blue light sensitive probe whose expression is targeted to a sparse set of newly formed spines and recently potentiated synapses. And upon prolonged exposure to blue light, the probe changes conformation, initiating the RAC1 signaling cascade, and through interactions with the actin cytoskeleton, synapses expressing the probe shrink and collapse. And so we can effectively use it to selectively delete newly formed spines in this area. And there's kind of a schematic of this experiment. We treated the animals with chronic cort, spines disappeared. We treated them with ketamine, some of them came back. We then deleted those spines and asked what impact that had on behavior 2 days or 1 week later. And what we found, as expected, chronic cort increased immobility. Ketamine rescued that effect, but the effect wore off if you deleted the spines. We also had an important negative control experiment where we optically deleted spines 3 hours after the ketamine treatment. You'll recall I told you that there are no new spines related to ketamine treatment 3 hours after injection. And so anything that we're deleting here has nothing to do with ketamine. And we found that that had no effect. So together, what we think what this means is, these new synapses are important, not in the way we thought, not for initiating the effect, but rather for sustaining it. And other interventions aimed at boosting spine formation might be useful for extending the durability of ketamine's antidepressant effects. And that, of course, is a big challenge with ketamine. And so we think this might be useful. But of course, this also begged another question. And if these new spines aren't required for initiating the effect at the circuit level, what is? And so that's the topic of a whole series of experiments that we conducted. This was in collaboration with my colleague at Cornell, Josh Levitz, led by a co-mentored postdoc, Hermione Munguba, as well as other lab members, and Brian Kobilka and Evan O'Brien at Stanford. And one hint for how this might work comes from recent work at Stanford showing that if you, this Alan Schatzberg, Nolan Williams, if you co-treat patients with ketamine and naltrexone, they still dissociate, but they don't have the antidepressant effects. And so this led us to investigate whether mu-opioid receptor signaling might be involved in initiating ketamine's antidepressant effects. We found in kind of a million different ways, at least in the mouse, that it was. Here's one example. If you co-treat the animals with naloxone blocking mu signaling, you'll block ketamine's antidepressant effects. We also looked at whether ketamine might be serving as an agonist at the mu-opioid receptor, or alternatively, it might just be stimulating endogenous opioid signaling. We're kind of agnostic as to whether and to what extent those two mechanisms are involved. But to address that, we teamed up with Brian Kobilka and Evan O'Brien and performed an in vitro G protein coupling assay using purified mu-opioid receptors and GI-containing G protein heterotrimers. And we basically find that ketamine is kind of a modest partial agonist of the mu-opioid receptor. And so direct binding could be involved in this effect. It may also involve endogenous opioids. We next asked what cell types in the medial prefrontal cortex express a lot of mu, looking at various published and also new single-cell RNA sequencing data sets. We find that somatostatin interneurons in the medial prefrontal cortex express a lot of mu compared to other cells. And patching these cells, we find that mu activation hyperpolarizes these interneurons. And again, summarizing quite a lot of work, we did some photometry. We see this very rapid silencing of SST cells, like minutes after ketamine injection, wearing off after about 15 minutes. We also did some 2-photon imaging. We've looked at over 10,000 presynaptic boutons in vivo in these somatostatin cells. We see this rapid suppression and of signaling in these somatostatin interneurons, and interestingly, not going to show you here, but a reversal of chronic stress effects. So chronic stress leads to a hypertrophy of presynaptic boutons in these SST cells and enhanced activity, essentially. And consistent with the hypothesis, there might be kind of too much inhibitory drive to the projection neurons in the prefrontal cortex, and ketamine is undoing that effect. We went on to show that you can actually that mu signaling in these cells is both necessary and sufficient for driving the antidepressant-like behavioral effect in the TEL suspension test. One way of looking at this is by selectively interfering with GI signaling. Mu in SST cells is a GPCR that is coupled with GI proteins, as I said, silencing the SST cells when it's activated. And we can express PTX in these SST cells selectively, decoupling GI signaling from the receptor. And we see that blocks ketamine's antidepressant-like effects. Conversely, using photopharmacology tools that Josh Levitz's lab has developed, we can very selectively, with spatiotemporal precision, just turn on GI signaling just in this cell type, just in this brain region, and we see that just 15 minutes of GI activation in this cell type is sufficient to recapitulate ketamine's antidepressant effects on behavior. So we think that ketamine is initiating its antidepressant effects through GI signaling in these SST cells, and that in turn has led to kind of new insights about how we might leverage that mechanism to develop new antidepressants that share ketamine's efficacy without some of its side effects. Happy to talk about that further. In the interest of time, I'm going to move on. I want to shift gears and tell you a little bit about another unpublished story, and then I'm going to return to the question I posed at the beginning and some of the repeated imaging in humans. So another antidepressant that has emerged with a lot of promise in recent years is transcranial magnetic stimulation. And in particular, these rapid intermittent theta burst stimulation protocols that can be used to drive antidepressant effects in a matter of days. And we think now that this kind of treatment may engage some of the same antidepressant mechanisms that ketamine does. And it's also useful, we think, as a tool, kind of a probe for driving behavioral state switches, both in animals and in people. And understanding how it works for the purpose of developing new treatments, but also just using it as a tool to change the brain and to change behavior and to understand some of the questions I posed at the beginning. What are the mechanisms that are driving those switches? It's also an interesting treatment because there's a lot of theory out there about how TMS works, but it's hard in practice to develop data to really test those hypotheses directly in humans. So this is why we think a mouse model might be really useful. So this work, this is a collaboration with Scott Wilkie, Laura DiNardo, and an amazing MD PhD student they have there, Michael Gongwer, and 3 postdocs in my lab, Shane Johnson, Devin Rocks, and Laura Shallinson. We have been modeling TMS either using opsins or using kind of a miniaturized magnet. I'm going to tell you a little bit about the opsin work today. I'm happy to answer questions about the magnet work as well. But as I said, there are ideas that TMS might be working through potentiating synapses in local microcircuits, but it might also involve network level mechanisms. If you're stimulating this area of the prefrontal cortex, that might activate other brain regions that are connected to it. And there's some correlative evidence in humans that may be the case, but of course it's hard to unpack what's really going on here, what's required and what's just correlated. Another interesting thing about this is TMS has this very large parameter space that's just kind of waiting to be optimized, but it's hard to do that optimization in people. There are questions like what is the optimal anatomical target site, the pulse dose, the duty cycle, the duration of stimulation, how do you optimize for network engagement, what about the intersession interval for stimulation? All of these things are thought to matter, but it's not clear how to do them most effectively. And again, this is somewhere where we think animal models could be really helpful. And so what we set out to do first was essentially to model the SAINT protocol, which is developed by Nolan Williams. Many of you are probably familiar with it. Essentially condenses 6 weeks of treatment into a single day through 10 sessions of intermittent theta burst stimulation, and then repeats that 5 days over the course of a week. And they see these really amazing response rates, at least in some individuals. And so we tried to model this in animals using in mice using a fast acting opsin, Chronos, expressed in projection neurons in the prelimbic cortex. We validated electrophysiologically that they're able to spike with high fidelity in response to this very high frequency intermittent theta burst stimulation. We also see these dose-dependent antidepressant-like behavioral effects on a host of different assays. This is immobility in the forced swim test. This is latency to feed in the novelty suppressed feeding test. We've also looked at other behaviors. And consistent with the idea that potentiation of neurons in local microcircuits might be involved, we see that intermittent theta burst stimulation increases the CFOS response in prefrontal neurons to kind of a probe stimulation, and that continuous theta burst stimulation, which is thought to be inhibitory tends to do the opposite. But kind of interestingly, and again, not what we would have predicted, these trends are actually not significant in the left prelimbic cortex where we're stimulating, but they're much more pronounced in the contralateral hemisphere. I'm going to return to that in a moment. We also did some spatial transcriptomic profiling after this accelerated ITBS protocol. In the interest of time, I'm not going to delve into this. I'll just tell you that this work identified layer 2, 3, and layer 5 IT cells, a particular kind of projection neuron, as especially important in this antidepressant-like response, and identified a host of signaling pathways that might be leveraged, we think, to gain mechanistic insights, and perhaps also to think about like drugs that could be paired with TMS. But one of them involves the formation of new synapses, particularly in layer 5 IT cells, and we were able to confirm that that is indeed the case, that this ITBS protocol drives spine formation and synapse formation in these cells. We think one way this can be really useful, as I alluded to, is leveraging the tools that have been developed and are available in mice for probing network level mechanisms. And so we wanted to ask, just in an unbiased way, to start which brain regions are potentiated in response to this intermittent theta burst stimulation. And so we treated mice with this kind of SAINT type protocol or with an equivalent protocol involving 10 sessions of continuous theta burst, which is thought to be inhibitory, or a sham treatment. Essentially, these are mice not expressing a GFP, not an active chronos. And we then perfuse the mice 90 minutes after a probe stimulation session, and then we do shield tissue clearing and see phosphoimmunohistochemistry, light sheet microscopy for imaging the whole brain, and we can quantify which brain regions are showing a potentiated response to a probe stimulation in response to this treatment. And what we saw was really interesting in a number of ways. I'll just unpack a couple of them here. So this is a map of the brain areas that are potentiated in response to this treatment. And many of them are brain regions that we might expect to be involved in depression: the orbital frontal cortex, the anterior cingulate, the insula. Some of them are also areas that we don't typically think about in depression, and like primary sensory motor cortex. And we're looking more at what role that might be playing. But one of the most consistent areas, and we've replicated this multiple times now, that also shows the largest effect is the insula, both the posterior and anterior insula in the mouse. I'm going to return to that in a moment. This, by contrast, is the effect of a single session, essentially nothing. So this shows that repeated stimulation really is necessary. We're looking now at like how much repeated stimulation, but there are very few effects of a single session of ITBS. And interestingly, when we look at the effects of 30 sessions of continuous theta burst stimulation, we also see almost nothing. And this is not what I expected to observe, honestly. So just to contextualize things here, this is the exact same number of pulses over the same number of days and the same number of sessions. The only difference is this kind of intermittent versus continuous rhythm. And what we see is almost no potentiation throughout the brain. We do see a significant depotentiation in the prelimbic cortex, consistent with the immunohistochemistry data I showed you a few minutes ago. And the only area that stands out is this medullary reticular nucleus, which we also see, by the way, in the ITBS condition. Again, not what we hypothesized, but one interesting kind of point to speculate on here is that there's new interest in the field of neuromodulation research that looks at how TMS affects heart rate variability acutely. And there's this idea that heart rate variability, when it is modulated by stimulation of the prefrontal cortex, that might tell you that you're stimulating the right brain region. And these nuclei in the brainstem that are potentiated by both CTBS and ITBS are involved in controlling heart rate. So that's interesting and could be pursued further, perhaps. But again, like I said, the most consistent area was this insula potentiation, and so we wanted to ask whether it was actually necessary for driving the antidepressant-like behavioral effects. I'm going to skip this. This shows we did some photometry validation. We do see a robust real-time response in the insula to the ITBS stimulation in the prelimbic cortex. And in this next set of experiments, we could deliver DREADs to the insula and chemogenetically inhibit the insula response during the stimulation period. And then the day after the stimulation is over, ask whether blocking the insula's response blocked the antidepressant-like behavioral effects. And that's indeed what we observed. You see a reduction in immobility in the forced swim test after ITBS, but not if the ITBS is paired with insula inhibition. And so, we think that this suggests that the downstream effects on the insula, at least in the mouse, are very important. To validate that in humans, we have been working with Aaron Bowes at the University of Iowa and Corey Keller at Stanford, who have intracranial EEG recordings from patients getting TMS treatments. And we see that many of the same regions that are activated in our kind of mouse CFOS mapping also show acute responses in the human brain, including the anterior cingulate, the orbitofrontal cortex, and both the anterior and posterior insula. And then in patients who are actually getting the SAINT treatment, we hypothesize that increased functional connectivity between the target site and the insula might be correlated with a higher likelihood of responding if you think that downstream effects on the insula are important. And so we have this data set, again, in collaboration with Nolan Williams at Stanford, of several dozen patients who've gotten SAINT image before and after. We just asked whether connectivity between the insula and the target site was associated with treatment response, and we found that it was. This is a moderate correlation, but an effect size, honestly, that's about as high as I would expect in a study like this. And this gives you another way of looking at the data. This is the neuroanatomical distribution of the treatment predictive connectivity features across the whole brain. And what we see is that the most important area across the whole brain in predicting response lies in the insula, especially the anterior insula. So we think this is kind of validation of the utility of using this mouse model, and we're now looking at ways that we might optimize the delivery of TMS in humans to engage this particular target. Okay, doing good for time. I'm going to shift gears and tell you a little bit about our human work, and then I'll stop and there'll be plenty of time for questions. Okay. So on the human side, we've been very interested in understanding how imaging might be useful to get a better handle on the neurobiological basis of heterogeneity in depression. For clinicians in this room, this will be very familiar. We diagnose depression when a patient presents with five or more of these 9 symptoms. And that means, doing some quick math, there are at least 256 unique ways a patient can present and still get this diagnosis. But I think it kind of stands to reason that someone who's experiencing weight loss, profound insomnia, intense anxiety, and physical agitation, this person may not have the exact same biological problem as the person who gets the same diagnosis and has almost opposite symptoms, like gained 30 pounds, can't get out of bed, physically slowed, not agitated, not really anxious at all, just profoundly anhedonic. And so in a way, it's kind of a miracle that our treatments work as well as they do for these very different people. And one thing we've been trying to do is get a better handle on how we might subgroup patients and understand whether different mechanisms are involved in controlling kind of the emergence of depression in these different individuals. Of course, there's been quite a lot of really pioneering work in this area. Most of it, I would say, at least until 5 or 10 years ago, has tended to group patients based on their symptoms. And some of these kind of clinical subgroups have been very influential, like seasonal depression is one that's seeped into the public lexicon. We're all aware of it, especially this time of year as days are getting longer. And yet it's been challenging to take these clinical subtypes and use them to identify biological correlates that could be useful for diagnosing them or for predicting treatment response. And so our approach has been to flip this upside down and ask whether we can group people based on biology and whether those biological groups are associated with different clinical symptom profiles or treatment outcomes. And the biological measures that we use are these resting state fMRI scans, which quantify functional connectivity between different brain regions based on spontaneous fluctuations in the bold signal, correlations in these spontaneous fluctuations. This first study that we did in this area combined data from all of the people you see photographed here who were generous enough to share their data with me. This is work from 711 subjects, and that was used for kind of training and optimization to discover these subgroups, and an additional 477 to replicate some of those results. We use fMRI to build these maps, which look kind of reminiscent of the correlation matrices I showed you earlier in the mice. Warm colors denote pairs of brain regions that are strongly functionally coupled. Importantly, most of these brain regions have nothing to do with depression. And so if you cluster patients based on all of these connectivity features, there are over 30,000 unique connectivity features in this matrix. You might get clusters, but they'd probably have nothing to do with depression. And so we needed a way to reduce the dimensionality of this data and to focus on the features that matter most in depression. And the method we settled on was canonical correlation analysis, which is sort of analogous to PCA. It looks for linear combinations of connectivity features that explain variance in the connectivity matrix, but then subject to the additional constraint that they should also be correlated with linear combinations of clinical symptoms associated with differences in clinical symptoms. And we find in this data-driven analysis strong evidence of at least 2 brain behavior dimensions explaining individual differences in anhedonia on the one hand, and anxiety and insomnia on the other. There's also, as you can see here, evidence of overfitting in this model. This problem can be solved by introducing a stabilized bootstrapped feature selection procedure and L2 regularization in the CCA. Happy to talk about that if people are interested. But what this shows you is that in green, the test set, and in blue, the training set, you get relatively similar correlations that increase with the number of connectivity features that you include, but you can kind of eliminate this problem of overfitting. Katie Dunlap, who now has her own lab at the University of Toronto, has replicated this and kind of extended it. She's identified 3 brain behavior dimensions. I'm not going to talk to you about Katie's work today, but check it out. It's in biological psychiatry. Amanda Booch, who's a former postdoc and will be starting her own lab soon, used a similar approach in autism. Others have used this approach in depression, in schizophrenia, in autism. We think it holds a lot of promise. What I want to kind of conclude with in the last few minutes is not persuading you that this is like the only way to subtype patients with depression. We obviously don't think that's the case, but rather that this particular solution is useful and that the approach in general will be helpful going forward. So we did hierarchical clustering on these 2 dimensions, identified 4 subgroups of patients that were associated with different kinds of abnormal connectivity. I'm not going to go into the details, but these different subtypes were also associated with different kinds of symptoms. Like this one had high levels of anxiety and anhedonia. Subtype 2 was associated more with low energy and fatigue. But perhaps most importantly for this conversation, we also find that they're associated with differences in treatment outcomes. This was inspired by this figure, which shows you the neuroanatomical distribution of the connectivity features that differentiate the subtypes. And one of them that was kind of most different across the subtypes was this area of the dorsomedial prefrontal cortex, which we know is a target for transcranial magnetic stimulation in depression. We also know from others' work that connectivity seeded from the target is at least modestly correlated with your likelihood of responding. And so we hypothesized that these people with very different connectivity might respond differently to TMS, and we found that they did. Subtype 1 was very responsive. Subtypes 2 and 4 were very unresponsive. Subtype 3 showed this kind of intermediate level of responsiveness. And we could actually train statistical classifiers to predict who would respond and who would not, based solely on connectivity features or a combination of connectivity features and subtype diagnosis, which resulted in an improvement in the classification rate. We can also do this just with clinical symptoms, but not as well. I hasten to add here that this is leave one out cross-validation. So I think the actual numbers here are not so important. They're likely to perform worse in a community sample. The point instead is that this is much higher than you'd expect by chance, and that this approach is probably going to be useful for understanding who's going to get better in response to TMS and who won't. And it needs to be tested, and that's what we're doing now. We have neuroimaging biomarkers similar to what I just described for predicting response to TMS at 2 different target regions, the left dorsolateral prefrontal cortex or the dorsomedial prefrontal cortex. You see there are a big range of responses to these treatments. Some patients do really well, some do not do well at all. And if you're a patient or a doctor, that's frustrating. If you're a statistician, this is an opportunity for developing models that can impact care. And so we think that by randomizing people to get the treatment we think will work better for them or the treatment we think won't work as well, we'll be able to not only evaluate the performance of the model, but also show how useful it is for improving care. Because there are situations like this one where if your response to one treatment was really correlated with your response to the other treatment, it wouldn't really matter how well the model performed. You could just roll the dice and give them one or the other, and they'll do just as well. On the other hand, if everyone's a responder to one treatment and a non-responder to the other, you can get everyone better if you just knew which treatment to give them. And of course, our modeling suggests reality is kind of this in-between scenario, and that these people in the upper left and the lower right are the ones we're really going to be able to help. And so we're testing that, and that should be done in about a year and a half. We're 200 patients in. I'll just briefly tell you about this work, and I'm going to kind of jump through some slides here so that there's plenty of time to ask questions. So I'm going to close by circling back to the question I asked at the beginning about repeated imaging and understanding the mechanisms that control mood state transitions. This is the work that Andrew alluded to. It's published, so you can check it out. I want to just mostly save time for discussion, but Chuck Lynch, who now has his own lab at Cornell, and Emmanuel Elbow, the associate director of our interventional psychiatry program, they did repeated imaging 40, 60, up to 80 times per person in a handful of patients as they cycle in and out of depression, and asked what's changing in the brain. I'm going to skip this, but it also allowed them to kind of map these resting state networks very precisely. And what they found was that one network, and only one network, was really dramatically expanded in the brains of patients with treatment-resistant depression. And that's this frontostriatal salience network, which occupies the anterior cingulate cortex, the anterior insula, and aspects of the lateral prefrontal cortex, as well as the ventral striatum. And it's markedly expanded in patients with depression. This is one individual. This is kind of a group level representation. It's an enormous effect size. We're used to kind of effects on the order of 0.2 Cohen's d. This is an order of magnitude higher, very replicable. You can actually diagnose depression based on nothing other than the topography of these networks fairly reliably. Going to skip this as well and close with this. And so we find, interestingly, contrary to our hypothesis again, that this network really does not change. It does not expand and contract as a person gets depressed and not depressed. It's really kind of rock solid stable, actually. You can see in these 4 examples from one person scanned many times, the precise boundaries really don't change. It's unrelated to how depressed you are now. It's unrelated to how many depressive episodes you had in the past. And that led us to kind of rethink what this network expansion might mean. Perhaps it's not driving a mood state switch, but is rather kind of a marker of risk for becoming depressed in response to, depression triggers in your life. And so to test that, we thought about different ways that we could approach it, and we turned to the ABCD sample, which scanned 10,000 kids. And among those 10,000 kids scanned at ages 10 and 12, we identified 60 that were never depressed at ages 10 and 12 and later became depressed in their teenage years. And the hypothesis was that this network would be expanded in those kids who went on to become depressed before they even got depressed, compared to kids who didn't. And that's indeed what we found. The effect size was smaller than in our treatment-resistant depression patients, but still quite significant. And then interestingly, and this is the last piece, we find that although the network isn't expanding and contracting, connectivity between nodes of the network is quite dynamic. And one set of connections in particular between the anterior cingulate and the nucleus accumbens. And you'll recall that in our mouse, we find that accumbens projecting cingulate cells encode this reward predictive signal that scales with effort and reinforces kind of effortful reward seeking decisions. We find that connectivity between these areas is quite correlated with how anhedonic you are today, not with other symptoms. And interestingly, it's predictive of how anhedonic you will be at your next scanning session before you even become anhedonic, consistent with a role in actually driving the effect and perhaps being a good treatment target. And that's something we're looking at as well. And I'll stop with that. I just want to thank all the people in red who did this work, as well as my collaborators and funding support. And we can take some questions. Gosh.
Yeah, it was a great talk. So much to do on it. I mean, I have many questions, but maybe one of them is like thinking about, integrating some of this like network level findings relating the anterior insula and the and prefrontal regions, in recovery from depression with some of the cellular work, thinking about like IT cells and thinking about spines. And I don't know if you guys are, looking at whether or not, obviously there's bidirectional connectivity, and these IT cells are a good substrate for that. So I don't know if you guys are looking into like, are there, specific cellular substrates, either, anterior insula, connections onto specific spines on specific, subtypes of prefrontal neurons, or, is there enrichment for prefrontal neurons which project to, the insula, or, also like somatostatin interneurons, are they getting preferential input from the insula? There's a lot of different themes to connect that interesting network level finding about insula and prefrontal regions with these microcircuit level findings about layer 2, layer 2, 3, and layer 5 IT cells, somatostatin neurons, and spines. And I don't know if any of those connections are currently kind of panning out or things you're looking at.
Yeah, great question. Yeah, like we're interested in most of what you just said. We've been thinking about. We've done a little bit of work on some of those questions. Why are these anterior insula cells important? We do it, like many people have shown that, like, if you were to rank the top 3 most consistently implicated brain regions in depression across thousands of neuroimaging studies, pretty clearly the anterior insula, the anterior cingulate, and the hippocampus would be those areas. And the anterior insula may be the number 1. We know that the anterior insula is involved in encoding interoceptive information. You can imagine how representations of the body's kind of interoceptive state might be important in modulating motivation and effortful reward seeking based on other kinds of drives. So we've been trying to understand what these cells in the anterior insula are doing behaviorally, and whether those that are involved in interesting behaviors in the context of depression are modulated or preferentially modulated by the prelimbic stimulation. We've also been looking, apropos of your question about the IT cells and whether particular projections are preferentially engaged by the stimulation. One question you might ask is whether that map I showed you of the distribution of brain regions that are potentiated by the prelimbic ITBS is explained by which regions are connected to the prelimbic cortex. And the answer is sort of interesting and surprising to us. Like, it is, but not really. So like the correlation is quite low. It's like 0.2 or something like that. And it suggests that the mechanism probably does involve direct projections from prelimbic to these brain areas. We find that, but like many areas that are strongly connected to prelimbic don't get potentiated, and others that are only weakly connected do get potentiated. And so there are probably kind of complicated network level effects that are determining, why some areas get potentiated and why others don't. And we're trying to probe that further as well. Yeah, we could talk further afterwards. Yes.
TMS activates both the neurons, GABAergic neurons, as well as glutamatergic neurons with lower threshold probably for the interneurons, just because they're smaller. The model, basically, you used CAMK2 promoter, so I'm assuming it activates mostly glutamatergic neurons. And so the question is, do you see a similar effect when you activate everything like we do in TMS clinically?
Yep. Yep. Great question. And you might ask the related question of, if you stimulate magnetically instead of using opsins, do you see similar effects? And the answer to both is yes. Scott Wilkie's study actually it's all a miniaturized magnet, kind of a mouse compatible TMS magnet, and it's up on bioRxiv, and I'd refer you to his beautiful work on that. As to the question of the degree to which our optogenetic findings resemble what actually happens with the magnets, they look quite similar, and they look similar in our hands as well. We think, both approaches have pros and cons, and one of the pros of the optogenetic approach is that it allows us to ask questions like the one you just posed, whether particular cell types are, whether and to what extent particular cell types are involved in different kinds of behavioral effects. And so we find that, I showed you CAMK2, we find that doing this on a synapsin promoter has a similar effect. And we found that just stimulating the insula projecting prelimbic cells also has essentially the same effect. That suggests that, on the one hand, that the engagement of these insula projecting prefrontal cells is sufficient to drive the behavioral effect on this particular behavior. On the other, many other cells are also engaged, and we're not making the claim that they're unimportant, but we think that this approach will, kind of allow us to unpack, hopefully, different roles for these different cell types, and that's something we're working on.
I have a follow-up question, if you don't mind. Sorry, just not directly related to this one, but you showed very nicely like a clinical correlation between the TMS effects and the connectivity with the anterior insula.
Yep.
And you also showed a strong correlation from the ABCD study between the ACC and the ventral striatum. But I was curious about the direct relationship, if there is any, between the anterior insula and the ventral striatum and the accumbens. Have you looked at that and its role in behavior and mood in general?
Yes, love that question too. Caveat, we're a little limited in the claims we can make based on the data we have, because they're mostly clinician rated scales or subjective self-reports, not behaviors. But it does look like there are some interesting differences. So what I showed you was this connectivity between anterior cingulate and ventral striatum was associated with anhedonia, but not with anxiety. And we find that connectivity between the anterior insula and the ventral striatum is associated with anxiety, but not with anhedonia. Another interesting feature is that, which I didn't show you, is that these correlations are much stronger in some individuals than in others, much more than you'd expect by chance based on kind of the confidence interval on the correlation coefficient. So we think that this different mechanisms might be involved in different people, but on average, we do see this double dissociation. Yeah, anxiety.
Thank you very much. Fantastic.
Thanks, everybody.